Can you see me now?
No.
No.
Oh, I don't know why. This is very strange because the camera is on.
Relog then. Log in again.
I did that.
Yeah, still it is not coming.
Oh, that's very strange. Okay, let me try to get into a different LAN. Give me a couple of minutes. I will try and see what I can do.
Why now?
Hello, can you see me now?
No.
Very strange because the video is on and I don't understand. So I'm not at all sure what to do, but I can give the talk. Let me share my screen and see whether that comes on. Okay? Can you see my screen?
Yes.
Okay, that's good enough. It's fine.
That's good.
That's fine. I'm sorry, but yesterday it worked, right? So I have no clue why it's not working today. Yeah.
Short while our registrar is joining, she will address and then we'll begin the program.
Yes. Right. I'm not sure what else to do.
One second, I will make her co-host and she'll speak and then we'll begin the program. Ma'am, can you, Naseem ma'am, can you on your camera? Yeah, right. So good morning. I welcome the resource person and the dignitaries on online platform for popular lecture supported by Indian Women Scientists Association, Navi Mumbai. Today's speaker is Professor Rohini Balakrishnan from Indian Institute of Science, Bangalore. So, ma'am will be addressing about the acoustics communication in between the animals. So, this is a proud moment for us to have the eminent personalities from Baba Atomic Research Center, Mumbai, Professor Lavinda ma'am, Professor Susan ma'am, and our Dean and Professor of Botany, Aruna ma'am, and we welcome our Honorable Registrar ma'am to address inaugural address for this gathering. Over to Naseem ma'am.
Thank you, Dr. Halim Khan, sir. Our esteemed eminent scientist speaker, resource person, Dr. Rohini Balakrishnan, madam, senior professor and chairperson of Center for Ecological Sciences, Indian Institute of Science, in popular science lecture through virtual mode organized by Indian Women Scientists Association, Navi Mumbai, in association with Department of Botany of Telangana University, distinguished Dr. Lalita Darveshwar, madam, President of Indian Women Scientists Association and former head Laser Physics Division, Baba Atomic Research Center, Mumbai, and Professor Suzanne Ipan, madam, former head Plant Biotechnology and Secondary Product Section from Baba Atomic Research Center. Convener of this event, Dr. Halim Khan, sir, Board of Studies Chairperson, Department of Botany, Telangana University, Professor Vidyavardhini, madam, Head Department of Botany, Telangana University, and Dean CDC, Professor M. Aruna, madam, Principal of College of Education, Tarangpur Campus of Telangana University, staff, students of Botany Department, Telangana University, delegates, and participants. Good morning to all of you. So, at the outset, I congratulate Dr. Halim Khan, sir, for organizing these events every month. And I think in this month, it will be, at present, it will be the 3 events. One, you have already organized Women's Day with very good personalities. And even today, you are organized this program with a very eminent scientist. And again, on Monday, that is 15th of this month, you are going to organize refresher course in collaboration with the head of the Department of Botany of Osmania University. So I appreciate your efforts and I congratulate you for this. Successfully, you are organizing events one by one. I think in each and every month, you are organizing maybe 2 or 3 programs. So I congratulate you. I'm sure that this program will be an opportunity for all the participants to meet and discuss the recent innovative methods in the concerned field. Deliberations, discussions, and exchange of views would be constructive, useful, and motivating by the eminent women scientists who are participating, who are the resource persons of this program. I hope it will render fruitful results. I wish this event a grand success. Thank you.
Thank you, ma'am. Thank you very much. Now, I will like to speak few words from our Dean and Professor of Botany, Aruna ma'am. Aruna ma'am, can you address us? Formally, you can address.
Yes, sir. Good morning, sir. Am I audible, sir?
Yes, you're audible.
Distinguished guests, respected Registrar of Telangana University, Professor Naseem madam, eminent scientists, Professor Lalita Dareshwar madam, who's president of Indian Women Scientists Association and also former head of physics from BAC, and Professor Susan, who's active member of Indian Women Scientists Association, and she's the one who has made us organize this program today. And we are thankful to you, madam, for your kind support. And Professor Rohini Balakrishnan, madam, who is resource person of today's program, and she's from Center for Ecological Sciences, Indian Institute of Sciences, Bangalore, and all faculty members from various universities and various disciplines, research scholars, students. Very good morning to you all. And I welcome you all to this popular science lecture organized by Indian Women Scientists Association, sponsored by BRNS DAE in association with Department of Botany, Telangana University, and we, the Department of Botany from Telangana University, are organizing many more or as such as programs where all the students, all the scholars of not only our department, but from various other universities have been benefited. And with these few words, I welcome you all to this popular science lecture being organized today. Hope that all the participants will be with us today till the end of the program and be benefited. And we are eagerly waiting for you to start the program. And we would like to listen to all your inspirational lectures. And thank you. Thank you, sir, for giving me this chance. Thank you.
Thank you, ma'am. This was our Dean, the Faculty of Science and Computer Science Engineering, Professor M. Aruna, ma'am. So over to Lalita, ma'am. Lalita, ma'am, shall I start sharing the screen, or you will be sharing?
Yes, I'll share the screen. So first of all, thank you very much, Professor.
Ma'am, wait a minute. Now we are going to have the introduction to Indian Women Science Association, Navi Mumbai, by Professor Lalita Dhareshwar, ma'am. She was former head for Laser Physics Division, Baba Atomic Research Center, Mumbai. Over to Lalita, ma'am.
Yes, thank you so much, Dr. Ahmed, and thank you very much, Professor Nasima and Dr. Aruna, and also our today's esteemed speaker, Dr. Rohini. I would like to share my screen now and introduce you briefly to our organization, which is very unique in the country. So let me just go over to my, are you able to see my screen? Are you able to see my?
Yes, your screen is visible.
Yeah. So this is, our Indian Women Scientists Association, which you are able to see here. We call it abbreviation of IWSA, IWSA. And the imposing building that you see here, the 4-storied building, is a working women's hostel. And adjoining that perpendicularly is our activity block. So this entire building was actually came up in it was the Indian Women Scientists Association was actually registered in 1973, and the building came up in the late 1980s. And the purpose of this entire work was to bring women scientists, more women in the field of science, and introduce to the world, the women who are actually working in the field of science, and empower them. And alongside to this, you are seeing some very beautiful pictures of some flora here. And these are the pictures of our new initiative, which is called the Learning Garden, the open air laboratory. And since you are all botany students, you will be very much appreciative of this. We have about 500 species of various kinds of plants on our premises, and 70 sections of these are divided into sections, and we are trying to bring about learning, science learning, through this learning garden, which is and we call it the open air laboratory. Now let me go ahead and give you the introduction in a nutshell. It has our organization, which is a non-political, non-profit organization, which has completed 47 years. It was founded by 12 committed women who were in very prominent positions, like Padma Bhushan, Professor Kamal Ranadive, who was an oncologist, and the Padma Shri, Dr. Shanta Gandhi, and many such women who came up with the idea of starting an organization to bring more women in the field of science. So our headquarters is situated in Vashi, Navi Mumbai, and we have 11 branches across the country. And you will see on the right-hand side the names of the branches and the year of their starting. Roorkee was started in 1979, Hyderabad, Pune, Nagpur, Kolhapur, Delhi, Kalpakkam, Baroda, Amravati, Bangalore, and Nellore. Bangalore and Nellore are the most recent branches. And we have more than 2,500 members. And our main mandate is to take science to society and women empowerment. And broadly speaking, we have two arms to our work. One is based on community welfare. The other one is the science awareness. And in 2019-20 alone, in 1 year, we have reached out to 23,000 recipients through our various programs. You can see here, under the community welfare activities, we have a working women hostel, we have a healthcare center, computer center, library, daycare center, nursery school, early childhood care education program, scholarship awards. Those boxes which are in green are the ones which stayed on even during the pandemic period. They were not affected. In the sense, the working women's hostel still kept our, the girls, working women who were not able to go back to their hometowns, and they stayed back, and we looked after them during this entire period of 1 year. And now slowly it is picking up, and this hostel is having a total capacity of about 160 beds. And we have a daycare center which is unfortunately not open now. And this is basically to cater to the children in and around the Navi Mumbai, from 6-month-old to 13-year-old children. And they are taken care with utmost care, affection, and love. And also at the same time, kind of infusing them with all the qualities which are required to pursue a scientific career right from the very young age. Then we have a nursery school also, which is also taken, about 36 children come to this nursery school, but it is at present not working. We have a healthcare center with a dentist and a physiotherapist, and we have very recently opened the healthcare center with the physiotherapist. We have a computer center which is taking care of various kinds of classes for various age groups, right from the school-going children to senior citizens. And it is also having a MSc IT learning center here. We have a library attached to the Maharashtra government, and this has more than 6,000 books, science and other books also. And we have every year scholarship awards given to deserving merit cum means students. The ECC program is one of our most important program, which started 25 years ago and offers 1-year diploma in early childhood care and education, and affiliated to the SNDT Women's University. Even before the NEP 2020 recognized the importance of ECCE, where the children, very young children in the age group of 3 to 8 years, where 85% of the brain development takes place in this age group, how important it is to have teaching methodology and formalizing this, teaching methodologies for this age group of children. Even before NEP came into, started this, we already recognized this 25 years ago and started this particular course, and where 600 to 700 women have passed out and have been placed well in many schools, and they are doing very well. At present, because of a pandemic, all our courses are going on online, and we have 2 courses called Shadow Teacher Training Course and Understanding Learning Disability, which are very, very important from NEP 2020, where all inclusive education is being advocated very strongly. So how to kind of take care of children who have learning disabilities, how to bring them into the fold, general fold, how to make them fit for the society once they come out of the school. All these things have been thought of, and these courses are being offered to people so that our society becomes more inclusive. Now, science education initiatives include school level as well as college level students. At the school level, we have a small science laboratory and a science library, and we have what is known as a science nurture initiative, where, as you see on the right-hand side, children from 7th and 8th standard are taken into our fold. They come every day to our premises. They spend 2 hours with us after the school. So they are offered lunch, and after lunch they sit and study with the teachers, and they learn science, maths, English, as well as computer skills. And these children are basically from economically deprived section of society. And by the time in 2 years, when they go out from our premises, they become so strong and motivated that even when they came, they didn't know how to even speak and speak in a coherent manner. But when they leave us, they become so empowered that they are able to do even a small, research, even on a small topic, and present it to the teachers, which is a great achievement. And for about 10 students or 15 students, we have 10 to 15 teachers. So you can see the ratio is so high. Teacher to student ratio is so high, which can never be there in any school or any college. In addition to this, we hold science lectures, science camps, science exhibitions for the school students. And for college students, we hold regularly popular science lecture series, which you are right now attending. And we also organize lecture workshops or based on certain themes, like science academies with the academies support, the 3 academies in India. And we hold refresher courses for the teachers also. Here you can see the college students having a look at a model from Department of Atomic Energy on a pressurized heavy water reactor. So recently in January, we had a science exhibition where DAE has exhibited several models on the projects that they are doing presently on various types of special materials and reactor grade materials for various other applications. So this is the kind of science education initiatives that we have. And in addition to these initiatives, now during the COVID last 1 year, we started several online webinar series. And we recently celebrated the Science Day online by holding science exhibition for the school students, where the theme was scientists, technologies, and innovators, which was aligned with this year's theme on STI. So under this, there were 3 categories. The children from 7th standard to 11th standard, they submitted working models. They had presentations of their favorite scientists. They had designed computer apps. And it was a real pleasure to see these children presenting their models, presentations, and computer apps in so much of confidence. And, it was really a wonderful webinar that we had in the recent times. In addition to that, we have what is known. There are 2 webinar series. One is called Member Enrichment Program, where our members, as you know, are all scientists from various backgrounds, from biology to computers to chemistry, physics, mathematics, artificial intelligence, name it, and you have them. And it's very essential that we have to share amongst us the knowledge that we have with the other members of our organization. So with this intention, we are organizing every week a webinar for our own members, where each member talks about her own in her own area of expertise. Like as you see here on online on microbiome overview, human and plant health, and on maximum murmuration, schooling, swarming lessons, and so on and so forth. And in addition to this, we have in the BRNS lecture series, which you are presently attending, our total outreach has been 3862 up to July 2020, but I'm sure now it has increased because we are having almost every week 2 to 3 lectures. You can see the various kinds of topics which are covered under this. We had laboratory experiments using computational modeling for chemical engineers, importance of quality of service in network for IT and computer, and biorefinery of seaweeds for biosciences. Then we had introduction to optical coherent communication for telecommunication engineers, and so on and so forth. And all our branches also at Kalpakam, Nagpur, Bengaluru, Roorkee, Delhi, they are also arranging so many wonderful such BRNS talks. You know, recently there was one by a lady who described her expedition to Antarctica, and it was something really a wonderful webinar. And in addition to this, now we have Science and Our Life series of lectures where a person who has a very high level of expertise in any field, it could be science, maths, health related, talks to general public. So it is bringing science to masses. It's literally that. So we had Dr. Nigamaja talking on healthy habits for bladder and bowels, Dr. Meera Venkatesh, who was in International Atomic Energy Agency for several years, who is also from BARC, spoke on applications of nuclear radiation for societal welfare. Then Professor Bindu Menon and Anita Pillai on artificial intelligence in healthcare, organ and tissue donation by Dr. Astrid, and the recent one, Professor Vidita Vaidya, on she spoke on not able to see that. She spoke on biology. I'm sorry, I'm not able to recollect the topic of her, but it was mostly on what gives rise to different types of diseases and changes in our at the DNA level due to constant exposure to stresses and strains, which are there in our daily life. Here you see Dr. Harish Mehta, a famous cardiologist, talking on the heart-related problems. Heart is your password to life. Various interventions which are needed at various times in our lives. And we had the good fortune of having Dr. Tessie Thomas as our speaker in this series, and she spoke on the aerospace journey. And today we are having, actually today I did not put the flyer, but Dr. Abha Nagral is going to speak to us on how to take care of our liver, which is a very important aspect in our lives, because liver is the most important organ, and it is if that gives way, then we have a lot of problems. So she's going to speak at 5 p.m. in the evening, and we will post that particular flyer and also the link to join the webinar in the chat box. I request one of our members to do that. And in addition to this, through our newsletters, which come every 4 months, we aspire to have knowledge sharing, bringing about a change, leading to value creation and promoting innovation with emphasis on science-based methods. Thank you very much for joining this webinar, because you being there, all of you participants, will really bring out the best from the speaker too. And please do join us and become a member of Indian Women Scientists Association. You will really enjoy being with us. Thank you. Now I will stop sharing and request Dr. Abdul to go ahead with this program.
Thank you so much, ma'am, for your wonderful introduction of the Indian Women Scientists Association. So recently we have celebrated Indian Women's Day, and it is a time that what women scientists are doing for community, society, and when they say being after the services also, they are rendering such a wonderful, they are shouldering wonderful societal work they are doing. So it is the metropolitan cities which are getting more benefit, but this is, I think, first time we are the people who are from the rural background got this opportunity. I would like to thank Susan ma'am, and over to Susan ma'am. Susan ma'am, can you start sharing, or shall I share that?
You please share, Mr. Khan.
Okay.
Yeah. Good morning, everybody. I am extremely delighted to introduce Professor Rohini Balakrishnan, PhD, Senior Professor and Chair, Center for Ecological Sciences, Indian Institute of Science, Bangalore.
One second, ma'am. 1 second. My PPT is opening. Yeah, it has opened.
Yeah. After doing her MSc, currently she is professor and chair of Center for Ecological Sciences, Indian Institute of Sciences, Bangalore. After doing her MSc in zoology, she did her PhD in biology with specialization in neurogenetics in 1991 from Tata Institute of Fundamental Research, Mumbai. Later, she went for postdoctoral research in bioacoustics in McGill University, Montreal, Canada, and again to University of Erlangen, Germany in 1996 to 98. After returning to India, she joined Indian Institute of Science in Bangalore in 1998 and still continues to work on acoustic communication in animals, focusing on crickets as a study system where she examines physiology and ecology of acoustical signal and reception. She is a pioneer in bioacoustics research in India and collaborates with mechanical and electrical engineers in interdisciplinary projects at the interface of biology and engineering. She has a large number of publications, 60 publications to her credit, with a H index of 17 with 711 citations. Her research interests focuses on animal behavior and bioacoustics, which include acoustic communication network, sensory ecology of mate finding and mate choice, predator-prey interaction, etc. She is also focusing her research on systematics and phylogenetics, which include species delimitation and identification, signal function and evolution, acoustic biodiversity monitoring, and soundscape. Today, she will speak on a very interesting topic, from crickets to elephants, the behavioral ecology of animal acoustic communication. Dr. Rohini, please.
Yes, thank you so much. Let me first thank the Indian Women's Science Association for inviting me. It is a privilege to be here and to speak to this audience of young scientists. I'd also like to thank the Telangana University and Dr. Ahmed for contacting me and enabling this. I'm sorry, I understand you cannot see me. It did work yesterday evening, but as long as the presentation can be seen and heard, I think that's important. At any point, in case either the visual or the audio disappears, please do alert me. I will share my screen now.
Yes, ma'am.
Yes. So can you
Yes.
see my screen? I will just put it in full screen. And yes.
Yes. Thank you.
All right, great. I'm going to also play out a number of audio files. So I am hoping, oh, I have to go back. I'm sorry. I did the sharing wrong. I will need to enable the, sorry about that. Okay, can you see my screen now?
Yes, ma'am.
Great. So I'm going to talk today a little bit about a variety. I'm going to give you a sort of very broad overview of some of the exciting animal sounds and questions that we have asked over the period of the last 20 years or so. And this goes all the way from small insects like crickets to elephants. I'm going to give you a little glimpse in honor of this being Women's Day, mostly of the work of women students. So I'm a person interested in bioacoustics and animal communication using sounds. And I'm interested in basically every aspect of it, from the structure and diversity to the perception and function of acoustic signals in an ecological and evolutionary context. As I said, today's talk is dedicated, coming just 1 week after Women's Day, to all the wonderful women postdocs and students who have been who worked with me over the past 20 years. And I hope, as these images will show you, a lot of these women have spent a number of months, if not years, out in the field, in forests, trying to get the data on acoustics for their PhDs and postdocs. I must emphasize, of course, that I've also had male students, almost an equal number. But interestingly, it's been the women who have gone out and done all the hard and dangerous field work in my lab. And so I use this as a chance to honor them and to pay tribute to them. So I am from the Indian Institute of Science, and my journey 20 years ago in bio time, just walking around the campus, trying to, can people see my cursor? Can you see my pointer or cursor? Otherwise I will.
Yeah.
You can,
Yes.
excellent,
Yes.
excellent. Let me use the pointer, right. So I would walk around the campus listening for these field crickets and recording their sounds. And what you see on the right is actually the 14 species or so that are actually right here on our campus. And so my early work started really just with insects on campus. And to tell you a little bit about the crickets, so male field crickets sing and they produce sounds by rubbing their forewings together. So they will typically sit in one place, maybe at the entrance to a burrow or in the leaf litter or in trees, wherever. But for a field cricket, it is usually on the ground and produce their songs. And females of their species who happen to be in the vicinity, if they hear these calls, will orient towards and approach males. You can tell that this is a female because she has an ovipositor, which is used for laying eggs. This behavior of males singing to attract females of their species, as you can see below, has been known actually for centuries. And this has been depicted in this wonderful painting from the 1700s by Rosenhoff. You can see a male cricket calling at the entrance of his burrow and a female cricket approaching him. And here is a rival male. So how do we know that the calls of these crickets actually attract females? The way that you can figure this out is if you can make a recording of the song of a cricket using a microphone and recorder, and then you can play back that call out in the field. And as you can see from the size of this loudspeaker, this was done a very long time ago. You will see that playing out the song of a cricket species attracts females of that species in the field. So these songs are sufficient to attract females. And this was one of the first evidences, indeed, that these songs are used as mate attraction signals. So when one of my first students, Natasha Matre, joined the lab, this is now 20 years ago, one of the first things we did was to make recordings of the field crickets on our campus. And what I've shown you over here is a depiction of these recordings. So if you go out into the field and you make a recording, you can both look and listen to these recordings. Let me play you the, I have to remove the pointer to be able to play. It's a little bit problematic. Okay, sorry. So let me play you these recordings. I'm going to play you the first cricket species. I hope you can hear it. In case you can't, please let me know. So that is one cricket species. Here's a second one. And here is a third species. I hope you could all hear the difference in the calls. As you can see, these three songs sound quite different from each other. And what is interesting is that these three species of crickets actually share the same patch of grass and call from there. You can look at these recordings. If you look at the top recording, you can see, what you can see is time on the x-axis and relative amplitude of sound on the y-axis. And what is very evident, of course, is that these songs are discontinuous. You could also hear it when I played the call. So you can hear the preep, 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 preep. So those preep, preep, preep are these chirps that you can now see when depicted as an oscillogram. Okay? But you can also see that these chirps are indeed made up of even smaller units called syllables. So there are two levels of structure here. There is the chirp, and the chirp itself is made up of smaller units called syllables. Each syllable is produced by one wing closure movement. What you can see if you look at these three depictions is that they differ both in the rate and duration of the chirps and in the rate and duration of the syllables. So basically, the rhythm parameters of these, the rhythms of these are different in different species, both at the level of the syllable and at the level of the chirps. And this is an important feature that females can use to distinguish the males of their own species. So if I take an analogy, if you were played 3 film songs, you already know immediately which film song is which. How come you know that? You know that because there are rhythm components that are different, but also because there are components in the pitch. So if I go to the right side, can people actually see my cursor on the screen? Or not? Because then I will have to go into using the pointer, which doesn't allow me to play. But anyway.
Yeah, you can see the cursor.
Oh, you can see the cursor. Okay, that's
Also,
great,
yeah.
because then I don't need to use the pointer. That will save me some time. Yeah. So if you look at the right panel, that consists of a depiction of the pitch of the call. So the pitch is the tone or the frequency of the calls as opposed to the rhythm. So what you see here is a power spectrum. On the x-axis, you've got the frequency of sound. And on the y-axis, you have the relative amplitude, how loud that frequency is. So what this power spectrum actually tells you is what is the frequency content or the pitch of the calls. And if you look at these 3 spectra, you can see that in each of them, most of the energy of this call is concentrated around one frequency, which is close to 5 kilohertz in 2 of the species. And the third species has a higher frequency around 7 kilohertz, which is why it sounded, you know, it sounds different, crank, crank, crank. That's because its carrier frequency is different. So between the rhythm or temporal features and the pitch, every species of cricket has a unique combination of these. And we know that crickets actually use these features to recognize their own species. How do we know that? So in one of the very first experiments that Natasha carried out here on campus, she dug a little pit in the ground out in the field, and she placed a loudspeaker inside it, and she played back the call of one of species from that loudspeaker. And then she asked, what does this female do? Basically, she just watched and she followed the female. And in those days, we didn't have any high-tech equipment. She would just watch, follow it. And once the animal had passed a particular point, she would put a little nail into the ground. And then in the day, she would come back and map out these nails. That's how these trajectories were made. And what you can see is that the female has walked relatively straight towards the trap as the call was playing, and then she will jump into it. So here's the track of one female. Here is the track of another individual female. She takes a slightly more meandering route, but then she ultimately finds this loudspeaker and jumps into the trap. So they will approach calls of their own species. Now comes the important thing. What if we play the calls of the other species that lives in the same area? Remember, there were 3 species. So this is a second species that calls at the same pitch, but with a different rhythm. So if you now in that same pit, through the speaker, play out the call of the second species to the female of species 1, what you see here, here's the release point, is that the female isn't interested at all. She walks away, in fact. The second female also, they generally wander around randomly, but they don't approach this other call, which they can actually hear perfectly well, especially because it's at the same pitch. So this shows you how you can use a behavioral experiment to demonstrate that animals can indeed distinguish between different kinds of signals based only upon their structure. You can do further experiments. So in this experiment, we changed only the rhythm parameters and kept the pitch constant. You can imagine now that you can do all kinds of experiments using this kind of method, which is you can change the pitch, but keep the structure the same and see what happens. Do they approach? Do they not approach? How often do they approach? And if you do a long series of such experiments, that gives you a very, very good window into how the brain of these creatures is actually processing that information, what features are these animals using of the sounds to figure out which calls are of their own species and which calls are of other species. But that was not our main question, because that had been done for decades. Many, many people before us had done that. But the problem that we were interested in really is what happens outside in natural environments, right? How do you locate one male when so many males are calling around you? There can be many, many males of your own species, of a female who's standing out there in the grass, and they are all calling together. And there are her own species and other species. Some are louder, some are closer, some are calling more often. Then there's wind. So how in this messy environment does a female actually manage to locate one male among simultaneously singing males? This is akin to what in acoustics is described as the cocktail party problem. So what is the cocktail party problem? So if you go to a party, any kind of party, lots of people, everyone's talking loudly. Now you are interested in listening to one conversation. You want to listen to one conversation in the room far away, and so many people are talking at the same time, but you're still able to do that. Okay, so how are we able to do that when everybody is talking at the same time? So this is a classic problem in acoustics. It's called the cocktail party problem. So the question that we asked is there a cocktail party? Do crickets have a cocktail party? That is, when there are so many different crickets calling together, is it going to be difficult to be able to figure out who is who? Is it going to be difficult to actually approach one single caller based upon their call? So to address this question, we first had to figure out, yes, is there a cocktail party? How do you figure it out for an animal like an insect? You have to figure out what is the acoustic environment that a female field cricket is exposed to out in the field. Sounds impossible, but you can do it. How can you do it? You need a number of pieces of information. So remember that the acoustic environment, that is of all of these songs coming together, basically depends on the singing males. So the first piece of information you really need to know is who are all these males who constitute the chorus of singers. And that is what is shown to you in this map. This is actually a map of the positions of individual male crickets calling in a field, and each letter represents one individual male. So basically what you have to do, you have to go in the field, you have to listen, you have to track down each calling male and flag that position, come back the next morning and map out in space where each of these animals is. So that's information piece number one. Second, you have to know whether these males move around. So how do you know that? And why do you have to know that? Because if they move around, the whole acoustic environment is going to change. This map is not going to be constant, right? So how you do that is in the course of a night, you go repeatedly back to each of these callers to see whether they are still in the same place and they are singing. So that's the second thing that Natasha did. In addition to mapping where animals are, she would note each time she looked at them whether they were singing, and also whether the same male was still where he was before. So that gives you information on how males are spaced, how often they're calling, and whether they're moving around. The second piece of information you need is what happens to the sound when it leaves the animal. How far away can the sound be heard? Okay. So to know that, what you need to do is to look at the loudness of the sound, which is what is mapped in decibels on the y-axis here, as a function of the distance from the cricket. So what you have to do is to get close to this cricket, have a sound level meter placed on the ground, and then slowly move backwards from there as the cricket is singing and see how that loudness decreases, which is the profile you can see here. As you can see and as you expect, as you move away from a cricket, as you move away from a sound source, the loudness will decrease. But what you can get here are the data on how it decreases as you move away in the natural environment along the ground from a number of different individual crickets. That is the profile that you have here. And as you can see, on average, that profile is quite similar across different individuals. But what we now have is how the sound attenuates as you move away. And as you can see, it goes below 30 decibels once you've got to about 2 meters away. But it goes below 40 decibels by the time you're about 1.5 meters away. Now, if you now then have information on the hearing sensitivity of the crickets, which you can do in independent lab experiments, you can figure this out using lab experiments where you play calls very, very softly and then slowly increase the amplitude and then see when the animals start to respond. So you get an estimate of where that lies, and that for this species lies along 40 dB. You can then use that graph to know at what distance a female will stop hearing the call, the average call of a male. You use that number to define the distance up to which the call of a male can be heard on average. And that is what this circle shows you. The circle shows you the average distance until which the call of a male can be heard. If these circles overlap a lot, what does that mean? For example, what it means if you look at A and B is that a female standing at this position will be able to hear A and B. A female standing at this position will be able to hear B, C, and D, and so on. So using these maps, you can now figure out how often these calls of different males overlap in time, how often a female is likely to hear 1, 2, 3 or more males, and that allows you to answer the question, is there a cocktail party? There is a cocktail party if a lot of the time a female actually can hear multiple males, and the answer to that question therefore is yes from our measurements. So these were our measurements to answer the question, is there a cocktail party? But is it a problem? Just because I'm hearing so many animals, does it make it difficult for me to find one? So to do that, you have to go back to doing an experiment. And we preferred to do that experiment in the field. And what we did was to have 4 speakers placed at a distance from a female placed here in the center. She's the little dot that you can see there. And we would play the calls of the species of cricket simultaneously from the 4 speakers. So it's like 4 males are singing together. And 3 of these males are at the same loudness, 76 dB, and one is half as loud, it's at 70 dB, which is at source. And now we ask, can the female find her way if we blast her with sound from all 4 speakers simultaneously? Can she find one of these speakers? And I'm going to show it to you in a video. This is filmed using an infrared camera. And here you can see the female moving. All 4 of the speakers are playing together. And you can see in this video, the path going toward one of them. And if you can see that little spot, the female moving, then you can see that she moves up to one of the speakers. So if you do this experiment again and again and again, so this is done in the field with a video camera mounted, then you can generate something like this. So what is this? This is simply showing you the paths of a large number of females who were tested on the same experiment. And as you can see from these parts, most of them reached some speaker or the other. 23 of them reached this speaker on the right, 5 of them reached the speaker on the left, 8 of them reached the speaker above here, and 2 of them actually turned around backwards and came down to reach the speaker below. So of 40 females, only 2 actually did not orient and reach a speaker, which means most of the time, even though they are being blasted by sound from 4 speakers together, they're actually able to find 1 speaker. And this is no mean feat. It's not an easy thing to do. So the next question we asked is, can we understand how they are managing to do this? Can we predict these scenarios? Can we understand how females are actually doing? So now we move from the field back into the lab. We have made our observations. We've done our experiments. The females have told us, come on, don't be silly. This is not a difficult problem for us, but actually it's a difficult problem. Because if you know acoustics and physics, then it's not an easy problem to solve. Okay, so how did we figure out how the females are managing to do this? The first thing is we already had all the data on the acoustic environment. We knew the positions of females and males. We knew how loud the sounds are. We know how they attenuate in space and the timings of these. So we could model these in a simulation, right? We can make a simulation of the scenario in a computer. And then we went to the literature to construct a virtual female cricket, or rather a virtual acoustic auditory system of the cricket. Why? Because there was a lot of literature. People had already previously worked on the ears of these crickets and how they work, and a little bit on how these sounds are processed within the auditory system of crickets. So we could use all of this information that was there, already published on crickets, and we could use all of that to sort of reconstruct a virtual female auditory system or a virtual female cricket. Now you release that virtual female cricket in this virtual acoustic environment, which reflects the actual acoustic environment outside. And you ask, what does this cricket do? You give it some rules, turn towards the loudest side, walk a few steps, listen again, and so on. So you can program such rules into them. And these rules were also based on our observations. And then you can now generate a set of simulated parts of these virtual crickets put under the exact same experimental conditions in the simulation as we had done in the. As you can see, just from a glance from these 2 figures, left and right, the real data on real females in the outside environment and the data on simulations of females who were placed in a replica of this environment in a simulation, just equipped with a few rules on how to process these sounds, we could actually recreate these parts. What does that mean? It tells us that the rules that we understand about the hearing and about how these sounds are processed in crickets are sufficient to allow us to understand how these crickets are able to orient towards these individual sound sources. This is, of course, somewhat complicated, but it's not necessary for us at this point to understand the complications in this. The fact is, it's possible to do it, and it tells us that we can use the rules of physiology that we understand to actually predict behavioral outcomes like this in the field. So I'm going to end over here to tell you a little bit about Natasha, who did this remarkable thesis on predicting acoustic orientation behavior in complex real-world environments. She never got as much credit as she should have for this piece of work. It's the first time anyone ever managed to do this in any real-world environment using acoustics. But she went on to do postdoctoral work on the mechanics of sound production and hearing in tree crickets at the University of Bristol with Daniel Robert, and we did this together. Basically, she looked at the biophysical mechanisms by which crickets actually manage to produce the sounds that they produce. And she's now a faculty member at Western University in Ontario in Canada. But there's much more to Natasha than that. She was an avid naturalist and photographer. And during her years here, she produced this wonderful coffee table book on the wildlife of our campus, which was so good that IWSA used this for its centenary publication when we celebrated our centenary, they released Natasha's book. She was also an avid snake enthusiast. She started Snake Rescue Volunteers of IWSA, and she headed a team that would go out and rescue snakes from people and people from snakes. So she was a person of many, many dimensions, my first student, and I'm very proud of her. I'm going to go on to the next piece of work that we did, which is another, again, large and interesting piece of work. And this, again, sort of asked a similar question, which is, if you have a sender, which in our case is a male cricket, produces a song, which is a signal, releases it into the medium, which can be air or water for crickets, it's air, and the intended receiver, if you like, is a female of his species. That's what a male wants when he sings. But when you're doing this outside, as I said before, you have to do it in the presence of a number of competing callers of many other different species as well. And this can result in a problem of masking interference. That is, everybody's calling together. Many, many species are calling together. And you still have to manage to get your signal across to the receiver that you want. The analogy that I will give you is cell phone networks. All of you use cell phones. You want to send a private message from your cell phone to the cell phone of somebody who you want to give a message to. You want to send a private message. But remember, you have to put that message out into a public medium. Again, the air, right? So your phone transmits this message that you have typed or that you speak into your device. It goes out as radio frequency waves into the air all over the place. Our atmosphere is filled with these waves. They're all, out there. They are clashing with each other, but they then have to be picked up by a single receiver, which is the phone of the person you are sending the message to. And that phone has to pick up only your message, not anybody else's message. Same problem, right? In communication in natural environments. And this is a problem that I have been studying for 15 years now. And I have been studying this problem in the context of an extremely complex acoustic environment, which is the dusk chorus. So all of you, I'm sure, would have noticed, just as you have a dawn chorus of birds, you have a dusk chorus of insects. As it gets dark, whether you're living in a city or you're living in a village or you're living in the forest, you will hear this dusk chorus, which is a large number of species, usually crickets, all calling together. If there are frogs, there will be frogs. So each of these guys is trying to send a message to a private receiver who is the female of the species. How do you do it? And there is this cacophony. What are the kinds of solutions that animals might use to minimize acoustic interference? Senders could use solutions and receivers can use solutions. In this talk, I'm only going to focus on the senders. What are the kinds of strategies that you as a sender could use to minimize interference and competition? One would be to partition your calls in time. For example, for these cricket species, since they use this in the context of breeding, if they are living together in the same habitat, then let's say this is species 1, species 2, species 3. At an extreme, species 1, 2, and 3 can breed at slightly different times so that they don't have to call at the same time. For a number of reasons, this can be quite difficult. Seasonality. In temperate regions, it's too cold and there's snow in winter and you can't do that. In tropical regions like ours, the monsoon often determines when you can breed and when you cannot breed. Sometimes it's too dry to breed, sometimes it's too wet to breed. So often many species will actually end up breeding at the same time. So this is a difficult solution. But within the same breeding season, even if you are calling together, species could partition their calling at different times of the night. For example, species 1 could call between, let's say, 7 to 7:30, and species 2 could call 7:30 to 8. So that way, if different species can partition their calling, then they can still call successfully and avoid interference. Or maybe they actually have to call at the same time. And like you said, the dusk chorus, most species like to call together. And then you can partition yourself at an even finer scale. So 2 minutes when somebody else is calling, I don't call when somebody else close to me is calling. And I put my call in the silent gaps, right? So you can partition in time. You can also partition yourself in space. So different species could call from different points In vertical space, for example, some species could call from the floor, some could call from the understory, some could call from the canopy of trees, and many species do. Okay, or you could partition yourself in acoustic space. What is this I'm showing you? I'm showing you a recording simply of one of these dusk choruses in a forest, and I have just visualized it, and you can see the time on this axis. What is on this axis is frequency, and you can see all these nice patterns over here. Now, each of these patterns is the call of one particular species, and you can see how beautifully segregated some of them are. So there's a species calling between 8 and 9 kilohertz, there's a species calling between 10 and 11 kilohertz, there's a species calling at 2 kilohertz. So just like radio frequency bands are partitioned when we communicate by radio, different species of crickets can also partition frequency space and call in narrow bands and tune in only to those bands. So all of these options are available. The question is, what is actually being used by these animals? And to find that out, you need a lot of information. You need to know who are all the species calling together in a community. You need to know what their signals look like, and you need to know when they are calling, where they are calling. And again, 20 years ago, we embarked upon this project in an extremely difficult environment, which was in a tropical rainforest. So a tropical rainforest is sort of the most complex acoustic environment you can think of. There are a large number of species calling together. And nobody had ever looked at such a complex environment before. Another of my first students, Swati Devakar, took upon herself when she joined my lab 20 years ago. And we decided to carry out the study in Kudremukh National Park in the Western Ghats, and you can see where that is. And you can see a picture of this. It's a difficult environment to work in, it's hilly, and all of our work has to be done at night. This is what a forest would look like if there's a nice moonlit night when you have to walk through it. This is what it will look like on a moonless night. You can basically see nothing. In addition, working in these forests at night had all kinds of other dangers. Lots of snakes, and Kudremukh has every poisonous snake you can think of, extremely poisonous snakes, including cobras, king cobras, vipers, etc. There are elephants, not at high densities, but they are there, so you have to constantly look out for them. At the time we were working, there were some Naxal problems, anti-Naxal forces going on, and that, of course, permits bureaucracy. Often bureaucracy will give you permits, revoke them, you don't know when you'll get them and when you can't. It was a very, very challenging environment, and I must tell you that in her very first field season, Swati got bitten by a viper. We spent 2-3 days, we spent 2-3 very anxious days in the ICU. She recovered from that, and I thought, never again am I going to send a student back into these forests. But Swati came back in 2 weeks and said, nothing doing, I'm going back. I want to do this, and I'm going to do my PhD on this. She was one of the bravest, most courageous students I've ever had. And just sheer grit and determination, she actually figured out who was this acoustic community, who was this community of insects. These are all small insects. You have to track each call, you have to make recordings, you have to catch the animal everywhere from the floor to the canopy of the forest. And within a couple of years, Swati actually managed to do this in our patch of forest. What did she find? We found 20 species of crickets, 10 belonging to one of the major groups, the Grylloidea, and 9 belonging to the other major group, which is the Tetigonioidea or Catidids, and 1 belonging to this interesting group called the Gryllacridoids. I'm going to play you some of these calls because they're really fun. So here there's a log cricket who lives on the floor of the forest. And this cricket, as with many other field crickets, as you can see in the spectrum here, most of his energy is at 4.5. What's very interesting about this cricket is that it will only call from rotten logs. Okay? So the rotten logs that fall to the floor of the forest are an extremely important calling site for him. This is another very interesting bush cricket with an incredible call. Lives on the forest floor, but also has spilled out of the forest You can hear it in many, many areas across southern India, actually also across northern India, different kind of species. And what's important about this call, as you see from the spectrum, is it's an extremely noisy call with lots of frequencies. Here's an understory cricket, which is probably a new species. Lovely call, just at 5 kilohertz. Another beautiful false leaf katydid. It looks like a leaf that Swati discovered in the understory. High frequency, 11 kilohertz call. My favorite animal lives in the canopy. Almost like a bird, calling at 3 kilohertz, lives high up in the canopy. And this, our most spectacular find, a waiter. So waders are these giant insects found mostly in Australia and New Zealand. And we discovered a calling species of this. I'm going to play you the cluck that you hear, like a frog. These also live high up in the tree canopy. Okay? So we have this very, very interesting set of species. And then we tried to test our hypotheses. Again, a grueling piece of work by Swati. She sampled transects in these forests in 3-hour periods around the clock. So we would do different 3-hour periods on different nights. And since she knew all the calls, she would count how many individuals of each species were actually calling, so that we could test whether different species were calling at different periods of the night. The result of her analysis, I'm not going to show you this because it's a very, very long story. But what we learned was, of course, the dusk chorus starts abruptly, dies off slowly after midnight. There's no dawn chorus of crickets in these forests. They don't call in the early morning. But the most important thing we learned was that there was no temporal partitioning of calling between these different species. Basically, they all like to call between dusk and midnight. Most of the species will not call after midnight. And even if you look, at much shorter time windows, you don't find very much evidence that they call at different times. So they're not using that axis of time. However, as I showed you before, we did find evidence for using different frequency bands in many of the species that we looked at, although there were some species that did not partition in spectral space. She then looked at spatial partitioning and asked whether animals call at different heights and at different strata. And yes, some of the species call only from the ground, some call a little above the ground, some call from the understory, and 5 of our species called entirely from the tree canopy. So they do achieve some separation based on space. But understanding pure physical space is not good enough. What we really needed to know was physical space in relation to acoustic space, similar to what I described to you in Natasha's work, but now in 3 dimensions. So we have male of species A calling here at a given loudness, and we have a male of species B calling at a different height in the chorus at a different loudness with a different call. And what you now need to know is how each of these calls attenuates within the forest. And in addition to knowing, of course, how far apart these animals are and how loudly they call. And this was the next piece of work that we undertook to figure out what happens to calls in the forest, these set of calls that we had discovered. This was done by my next PhD student, Manjari Jain, whose thesis was on forest acoustics and on what happens to signals in forests. And to do this, what she had to do was to take all of these calls of all the species that we had found, that Swati had found, and she played back these calls in the forest. So you can see these beautiful photographs showing you how hard this was for her to do. So she would have to walk kilometers into the forest with all of her equipment. In those days, the equipment were quite heavy. And then we would have to improvise and set up platforms for which we were helped hugely by our field assistant, Sudhakar. And then we would play back these calls. We would re-record them. We would look at their sound levels at different heights from the ground to the canopy. For the canopy, we got time bursts just to give you a flavor for what such data look like. So here is a ground-dwelling field cricket species similar to the one I played to you. And what you see here again is how the loudness of that species changes as you move away from the source, in this case, the loudspeaker. And you can see these profiles as you play at different heights. So this profile at red is when you play from the ground, and you can see how steeply it attenuates when you play from the ground. But when you play from above the ground, it is the attenuations are quite similar, and they're not as strong as when it is played from the ground. But the interesting thing is this animal calls from the ground. Okay? So it's not calling from an acoustically optimal place, and so on. So you can do these experiments as Manjari did, and it took her an entire 3-4 years to actually play all these calls from different heights in the forest for all of these species from all of the heights, and to look at what it does to these calls in terms of attenuation and the structure of these signals. Using that information, Manjari then went back to doing three-dimensional simulations. Again, very similar to the maps I showed you that Natasha had made. Each of these is, the center of each circle is the position of one caller of any given species. Now this is for multiple species. And the size of this circle, the call of this individual of this species will be heard by a female of that species. So these maps are similar to the maps I showed you in the beginning, except these are 3D, and therefore they were much harder to do, but we solved all the computational issues, and we were then able to compute if a female is standing at any random position over here, how often will she hear the calls of more than one male, including other species. When you perform this rather complex computation, which I'm not going to go into, this is the answer which we got, which is if you take all the axes of separation into account, the timing, the space, the attenuation, etc., the actual average masking probability is close to 0, and that's crazy. So basically our cacophony dissolved into silence. So our paper is entitled Cacophony or Sounds of Silence. So what I presented to you as that huge cacophony, you do all of those numbers and look at it through the of a cricket standing there actually isn't much interference at all. Okay, so it was quite a spectacular result that we got. So to tell you about Swati then went on to become assistant professor in the Department of Environmental Studies in Delhi University, where she is now, and she continues to do some amount of work on acoustic signals of crickets. She's focused on Northeast India, which is a completely unexplored area. Manjari Jain also went on to become an assistant professor. She's now an associate professor at IWSA Mohali, and she continues to work on the ecology and evolution of signals. She works on both crickets and birds, and she has a very large, as you can see, group of her own now. And it was my special privilege to meet both of these students at the International Bioacoustics meeting 3 years ago. And as you can see us here, both Manjari and Swati as proud professors with their own research groups, and I am very proud of both. Let me see how much time I have. I think I don't have too much time, so let me quickly go over the work of Samira. Samira Agnihotri, another of my students, who worked on vocal mimicry in a bird species, the greater racket-tailed drongo. She did her work again in the wild in the BRT, the Billigiri Rangan Hills in southern India, and you can see what those forests look like. What she basically did during her PhD was to follow individual drongos in the wild and make sound recordings of all their calls together with all what they were actually doing, so we could get the context. Let me quickly show you some sounds. So this is the call of a brown-cheeked fulvetta, a babbler species. And here is the mimicked call by the racket-tailed drongo. Again, a fairy bluebird, drongo mimicking it, the greater flameback woodpecker, drongo mimicking it. As you can see, they are very good mimics. And what you can see here are the visuals, the visual representation of those calls. End of this exercise, we identified that drongos mimicked 35 bird species, 3 mammals, at least 2 frogs, 1 insect. This is at the end of 2 years of fieldwork. So they do mimic a large variety, especially of bird species. But our question was, why? So to find out why, Samira carried out a number of playback experiments in the field. Basically, we would tie a speaker to a tree somewhere in the territory of a drongo, and we would play back drongo to them, and we were kind of so you get it? It repeats the same note over and over. And that kind of call, irrespective of this, in this particular set of notes, would attract. Oh, it's not stopped. Okay. So this kind of call would attract other drongos. So sometimes it could be male drongos or female drongos, but it basically often other male drongos who compete with it. A babbler species. So this is a babbler species that often forages together with drongos. So this babbler species, woodpeckers, there's a set of bird species that form what's called a mixed species flock in the forest. These birds forage together. And it seems that the drongo actually uses the calls of these other species to call them towards itself. It was crazy because when you play back these calls, these other species will also come towards it. And in Samira's observations also, when a drongo made these calls, these species would sometimes appear on the scene. So it's almost as if it was calling these birds to form this mixed species flock. So we got some very interesting insights into what this mimicry might mean for drongos. So Samira continues to study drongos and their vocal repertoires. She brought out a very, very nice CD at the end of her thesis, which was The Birds of Biligiri Rangaswamy Temple Tiger Reserve, a bilingual CD in Kannada and English, which talks of the calls of almost 150 bird species in this reserve. And that we need actually to put up on a website to become a good resource for people who would like to learn about bird calls. I know I'm almost over my time, but I think I'll just take 5 minutes to tell you a little bit about our elephant work and the fantastic work, the pioneering work that Smita Nair did on Asian elephants in Mudumalai. And this, of course, was together with Swati's work, one of the hardest pieces of work that we did. And although African elephants had been studied for a long time for their acoustic communication, nobody had worked on Indian elephants. And Smita was actually the first person who went out to try and document the calls of Asian elephants and what these elephants might be using them for. And as you can imagine, elephants are dangerous creatures, and this was hard and dangerous work. So I'm very quickly going to give you, she found 4 kinds of main call types of elephants. One, the very, very familiar trumpets. But she found some other interesting call types, roars. And this is a really weird call type. You'd never associate it with an elephant. It sounds like a squirrel or a bird, but it's an elephant. And then very famous rumbles, which are the very low frequency sounds that elephants use. So if you can hear something like a motorcycle, that's a rumble. And these are from African elephant studies. We know these are used for very long distance communication. I'm going to end with a couple of videos just for fun to show you the context of some of these. This was taken in Mudumalai. That's an aggressive trumpet chasing away wild dogs. Here's an example of a roar, which is often used in dominance interactions or between mothers and offspring in a herd. Probably a mother scolding her offspring. Here's rumble. As I said, this is for very long distance communication. And when I hope you can hear the rumble. Now it will start. So this elephant is producing a rumble, and it happened in a context when one of the herd members got separated from the herd. And that animal was also producing rumbles, and so were animals in this herd. And finally, strange and incident that we were lucky to be present at in Mudumalai. And I'll narrate this to you as it goes on, and you will hear some chirps happening. This happened when a group of elephants, probably by mistake, ran into a bear and her cub, both of whom climbed trees. And the elephants are below. The bear cub then came down from its tree, and you can see it coming down, and we thought the elephants would trample it. But you can see the bear managed to get onto the tree where its mother was. And the elephants are chirping in the background. Chirps are made when they are confused and alarmed. Right, so that's just to give you some feel. I'm ending now. As you can see, it takes a lot to study animal acoustic signaling. It takes a lot of patience because these are living animals and you cannot force them to do anything. If you're working outside, it often takes a lot of courage to be out in these environments, especially at night. It takes a lot of determination and persistence, but all of that comes if you have a lot of passion. And I think all of my students and postdocs have had this incredible passion to achieve so much. So once again, I end by sort of saluting all these women and many more, I'm sure, who will follow in their footsteps. And I'm open to take questions. Shall I share my screen? Hello, can you hear me?
Yes, yes.
Yes. Would you like me to unshare my screen? Right.
That was a wonderful talk, Mr. Rohini. Really so interesting.
Thank you. If there are questions, I'm very happy to take them from anyone.
Ma'am, are these signals have any purpose? Like in elephants, you have shown that mother and baby will have communication in some way. So what about in other animals? Like you have seen in birds, is there any kind of, particular, in what situation, what kind of signals they will communicate by the
That's a very broad question. And yes, even in birds in different species, you will find different kinds of signals when there's parent-offspring communication, sometimes different kinds of signals when there's aggression, different kinds of signals to attract a mate. With the insects, typically there are very few stereotyped signals, and these are specific to a species. And that's one of the reasons I find insects easier to study. But in things like drongos, elephants, most mammal species, there's very large repertoires. So you have to do a lot of work to figure out what signal is used for what function in what context. Yeah, so yes, there are different context-specific signals, but it's different for each species and each group.
What about other mammals? I mean to say, elephants are covered in the study. But what about the other mammals? There are a number of mammals, and they have a lot of parental care for their young ones.
Yes.
So whether such kind of any, I mean to say, characteristic study is done?
Yeah, not by me, but there's been a lot of work, for example, on primates, on bonnet macaques, on chimpanzees, on a lot of primates. People have done work. I mean, globally and even in India, some work has been done. I mean, you can't do everything, so you can. So birds, there's been a lot of work done. So yes, but one person cannot do. There's a limit to what you can do. But these have been studied, but there's still a lot to know. That's for sure. Are there any
participants? If anybody is interested in asking question, please you can give us raise the hand. I'll unmute you. Participants, among participants, if anybody is ready to ask questions.
Yeah, please do.
Raise the hand. I'll be making unmute to you.
Yeah, somebody has raised a hand.
Yeah, Paramjeet, can you talk? I have made you.
I can't hear.
Okay.
Hello, can you unmute yourself?
Or they could type the question as well. Then I can.
Or you can even type the question. If you want, you can live interact. Paramjeet. Any participant, please, if you want, you can put your words in chat box.
Please do. I'd love to hear your questions.
It's a rare time we are having an eminent speaker, and out of her busy schedule, yeah, ma'am has. Ma'am, are you able to see the question? Lalita ma'am has asked a question.
No. I wanted to only, in the end, show the today's evening talk by IWSA, under the Science and Our Life series, which at the end of the talk, I will just project that, if you don't mind.
Okay. Participants, please, if you are ready, we can ask a question. If not, we'll go for summing up. Arna ma'am.
Okay, now I'm able to unmute. First and foremost, congratulations, Dr. Rohini, for that excellent lecture. It was wonderful and very mesmerizing. You took us, on a journey of all the contributions of your students. It was so very motivating and inspiring. My question that I would like to ask is that are these acoustic signals also found in the prokaryotes? I mean, you have mentioned about the animals. I mean, say on bacteria, are these acoustic signals also working there?
I have no idea at all. I can't think immediately that the answer would be yes, but I think nature is full of surprises. And
yes, because we have done very preliminary
very close range mechanical interactions, which we have no clue about.
Yes, the thing is that we tried very preliminary experiments. I mean, now I'm retired, but in our college, there was a student who wanted to work on bacteria, and he was, also trying to play some music and see the, what is the effect of that music on the, growth rate of the organisms and the pigment production. But, we did not get much literature. So I was just wondering whether you would be able to, throw some light on that. So it would be really very useful to, guide them and to carry forward these experiments. But I mean, excellent. Your experimentation is just wonderful. Loved it. Thank you very much. Yeah.
Thank you. So just to respond to that, I know I'm not immediately aware of anything on any kind of, what one might call acoustic or even mechanical communication between bacteria. Chemical, of course, is very well known. Chemical communication is very well known. But, I think often we are limited by what we can measure. And at that level, we never had the tools to measure. Now, perhaps we have the tools to measure, but I am not at all sure, what kind of tools or what kind of signaling one should be looking for. And I'm sure those tools will have to be quite sophisticated for you to be able to pick up some signals. But no, I'm sorry, I can't.
So
chemicals definitely has been very well worked out.
Yes, chemotaxis is very, very known, very much known. But, because in the natural ecosystems, I mean, it is the whole biotic communities living together.
Yes.
Like, the insects, and then with the plants, we have the pollinators. I mean, everything, it's so closely connected. So I'm sure there would be, we just have to seek those answers probably.
You're right. I should tell you that very, very recently, in the last couple of years, people have figured out how plants actually can listen to the sounds of their pollinators, which was not something we would have thought about. So,
yes.
I have a small question. I'm Surekha Zingre.
Yes.
And I'm just wondering, do you require really very sophisticated instrumentation for recording? And second is, does the placement of these instruments, is it sensed by the crickets or the creatures whom you're recording from?
Yes.
Do they sense something different? Would the calls be different?
Yes. Yeah, two great questions. So the answer to the first is that it really depends what question you're trying to answer. If your question is simply about timing and rhythm components, you can do it with any kind of recorder. It doesn't matter because you're only interested in the timing of a signal. If you're working with tonal sounds that are in the range of human hearing, for which most acoustic devices are designed, then again you can go with very standard equipment. In fact, today many people are using cell phones, and there's no issue with that. But if you're working with things like ultrasound, if you're working with broadband signals, like with some of our insects that span the human hearing range and ultrasound, then you need specialized equipment. You need equipment that is specialized to cater to those frequency ranges. The second,
I was saying, do they sense the presence of, I should use the
observer.
word invader in their territory,
Yes.
and do they change their calls?
Yes. So this is something we have to keep in mind. And so one of the things we do is before we get out to start recordings, we often spend a lot of time watching and listening to these animals so that we know if we have disturbed them, because that is the first thing. You have to know whether they're being disturbed or not. Of course, if they're disturbed, most of them will just stop calling. But what one worries about is subtle changes in the structure. So that I think it comes with experience. We walk very carefully. We don't make any vibrations. We maintain our distance, the max distance that we can. And again, it's a naturalist sort of intuition that builds. When an animal is disturbed, if you have spent enough time with it. So one of the things I insist on with my students is that the first thing is always just go out in the field, be with the animals, track them, watch them. And unless you do this for a while, you won't know this. And this is important to know.
Yeah, thank you so much for this amazing lecture, right? From an orchestra to mimicry and then to these big elephants making rumbles and noises and roars. Thank you. It was really an eye-opener. Thank you so much.
Thank you very much. Students? So just a message to all the students here. I am really sad that this could not happen in person. And I think it would have been better if I could see you. But the message that I would like to send out is, I know, and I have been in that position as a very quiet student who never liked to speak up, because it is difficult to speak up when you are in an audience, though less so in an online audience, I think. But I think one of the things that all of you must try to do going forward is, don't worry about speaking up. If you have something to ask, to say, just say it. Even today, after 30 years, I ask completely meaningless and stupid questions. And I will give you the advice that was given to me by my mentor, Professor Sohan Modak in Pune University, where I did my master's. So he paraphrased, when he was my teacher, and he paraphrased an interesting saying in English, which is, I'm not quoting it exactly, but it is better to remain silent and be thought a fool than to open your mouth and be proved one. That is the saying. But Professor Modak said, for every student, the saying should be, it is better to open your mouth and have the chance to become less foolish than to stay silent and remain one. And that's always, I don't remember anything he taught me in terms of subject, but I remember this one sentence, and it has served me very well in my life. So I thought I'd share that with you.
Aruna ma'am, over to you.
Good afternoon, sir. Shall I sum up?
Yeah.
Good afternoon, everyone. On behalf of Department of Botany, we would like to thank all the resource persons and eminent scientists who were with us today for the popular science lecture organized by Indian Women Scientists Association in collaboration with BRNS and in association with Department of Botany. Firstly, I would like to thank our respected registrar, Naseem Madam, who has addressed the gathering, and she's one who is behind us in organizing the seminars from the Department of Botany. And we are thankful to you, Madam, for your encouragement and support. And we are extremely thankful to Professor Lalita Dhareshwar, Madam, who has introduced about activities of Indian Women Scientists Association. We are very proud to learn that Indian Women Science Association has completed 47 years, comprising of 2,500 members, with around 11 branches all across the country. And Madam has narrated all the activities of the association, and few of these are community welfare activities carried out at the association, where they work for women and childcare, and strongly involved in ECCE program. And it has started around 25 years ago, where many women have passed out and are placed at different positions. And it's very proud, and we are very happy to learn that this is working even during this pandemic period of current situation. And Madam has also told about a few other activities like science education initiatives, which have been carried out at school and college levels, and which include organizing workshops, refresher courses for faculty, and exhibiting for students' purpose. And also in addition to these initiatives, webinars were organized during pandemic period where all the faculty members and all the women exclusively were benefited. And out of this, there were around lecture series which were outreached, which account to around 3,862. And we are thankful to Susan Madam for introducing Dr. Rohini Balakrishnan Madam. We are thankful to Dr. Rohini Balakrishnan Madam, and thank you, Madam, for your wonderful presentation. And the topic of your presentation was From Crickets to Elephants, the Behavioral Ecology of Animal Acoustic Communication. Madam, it was really wonderful and amazing presentation where you have taken us from small insects like crickets to elephants. And you have dedicated your talk to all the women folk on their achievements during the celebrations of Women's Day. It's really grateful. And you have also taken examples of all your contributions of research scholars in your laboratory. And now they have been excelled and they are well placed as professors at different universities. And we are thankful to you, Madam, where you have enlightened the work about male crickets, how they sing to attract the females, which was really interesting. And acoustics made sufficient for the signaling. And you have explained the concept very well with the help of your audio. And you have also shown for recorded species specific songs like playing of insects at different levels of rhythms, which is a component of variation. And it's also learned that they do not move towards calls of other cricket species that is indicated saying that it is species specific. And one of the questions which was answered was really interesting, like, is there a cocktail party? The question to this answer was yes. And you have told all about the sensory ecology where it was discussed in an elaborate manner, and acoustic environment of a female field cricket in relation to outdoor phonotaxis experiments conducted in your laboratory was very well explained with the help of an audio. And as a resultant, we have learned that only females will run back, and correlation of females and acoustic environment was proved in all aspects of relative positions of females and males and song intensities and source. And you have also shared the work of all your scholars in the laboratory, for which we are grateful. And it was Natasha's work where she has done on mechanisms of sound production and hearing in insects. And she has interest in rescuing people from snakes. And she was a photographer. And it was all in natural environment, how communication is in relation to the signaling. And to explain this concept, you have taken us back with an example, making use of mobiles in our daily life, how signaling is correlated to that of communication. And Madam, you have also told about cacophony, dust chorus, and solution to acoustics interference, that is strategies of sender, and temporal partitioning in respect to seasonal, circadian, and fine temporal rhythmic specific to species, which was discussed. And with the help of a graph, you have sketched acoustic space in different species of crickets. And it was the work of Swati Divakar. She has worked on Kundarmarg National Parks. So slowly you have taken us to forests. It was really lively presentation. And you have narrated the work done by her. And she is a girl who remains a source of inspiration to all other research scholars who are really doing work in this field, because she had to walk, as you have told, she had to walk for a long time into miles of forest that too during night times. Hats off to you, Swati Divakar, and wish you all the best in your near future. And Madam has slowly switched on to biodiversity, where she had made us learn about different species. To quote few here are land river species, which is log cricket, which is only on rotten logs, and Mycopodia species, Biner, Broccopeplus, Westler, Grilocropsis. And we have learned all these species really new to us, Madam, all biodiversity, which was really interesting. We are grateful to you, Madam, and thank you so much. And it was later on switched on to work and contribution of Manjari Jain. She worked on forest acoustics and signal, as you have told, and she walked into forest with all the equipment which was needed, and you had to go for recording the program that is at levels of heights to take the sounds of all the crickets. That was really fascinating. And a 3D acoustic simulation model was also constructed, and it was Swati Manjaraji, now who is associate professor. So congratulations, wish you all the best. And it was Samira Agnihotri, who had worked on Rangan Hills in southern India, and she carried her work on Drongus. And it is learned that she worked on vocal mimicry, as you have told, Madam. And she has identified, that is, you people have identified mimicry of 35 birds and 3 mammals and so on. And syntax of mimicry says that repetitive notes will attract other drones and non-repetitive notes attract other species. So this is the resultant of her work. And it was Smita Nair, who has documented Asian elephant acoustic communication. And as you have told, we have learned that structurally classifying the mammals, 4 types were identified, like trumpets, roars, chirps, and rumbles. So here it was clearly explained how there is a communication between a mother and child, which was really interesting. And ultimately, Madam has ended her talk saying that the call of the wild will also need to have patience, courage, determination, and passion. So with these few words, I would like to thank Rohini Madam, who have given us an account on behavioral ecology, taking us from crickets to elephants. So in the present, on the current era of living world, we people in the society are finding it difficult in identifying the birds or in identifying the animals. So this is the way where each and every student or each and every scholar working in their field of research should have enough patience and work hard to achieve and to get the results. So nowadays students are entering into that field, that area of field where it has become easier to pursue their PhD. But seeing to your work where it's really lively presentation, all the scholars should really feel like working hard and give your presentation in near future. So you are a source of inspiration and it's really motivating. And thank you. And thank you all the members of Indian Science Association. And as Susan Madam has rightly pointed us, we have to take membership of that association, and we will definitely circulate it to all the women faculty of our university and other places. And we will become member if we are eligible. Thank you. Thank you, sir, for giving me this opportunity. Thank you all the participants who were with us today, right from morning to the end of the program. Thank you all.
Thank you very much.
Thank you, ma'am. Ma'am, Rohini, ma'am, one small query. You have gone in a deep greenery. So, were there any incidents that the scholars have failed? Any kind of had any experience of some kind of risk, like or any? Can you share the experience while live going in the wild area. How was that? Would you say?
No, like I said, if you're working in forested areas, then yes, I mean, you have to worry about snakes. You have to, and of course, you get bitten all the time by leeches and ticks and mosquitoes, which is actually the biggest, because that is there all the time. The other risks are much, I mean, the other risks are low. I mean, they're hard, but they're low. But in the rainy season, leeches, and the dry season, ticks and mosquitoes. So yeah, it does take a certain kind of person, I think, to want to do this kind of thing. The things that happened to Swati are a little, I always tell Swati, somehow all bad news comes to you because she got bitten by a snake, and then she had permit issues, and then issues with Naxals, and yeah, but it doesn't always all come together. And so it's not always like that. And there are many, many other kinds of projects where we work in agricultural fields. We do projects on campuses, and those are that way much safer in the sense that you don't have to deal with all these things. But, ultimately, for much of our work, we do at night. And so you have to be comfortable with working outside at night. Yeah. But the moment you step out of four walls, life is not the same. You have to be prepared for everything. And most importantly, dealing with people, which is not always easy on many, many different, we are working outside. If we are in areas which is like a village or something, 50 people will come to ask you what you're doing, and so you can't do your work. So there's all kinds of challenges, but it's really fun. And the students also, I think very importantly, and this is not relevant to the students who are listening to this, but students who come from urban areas, they have never lived in villages and things. And so it's a very good experience for them to, live in a village, know what things are like there, learn how they should adjust, and things like that. So I feel that when you do this kind of work, you get a double PhD. One is the academic, and the other is, an understanding of yourself and the country around you. And I think that's also very important.
Life PhD, yeah.
Yes, exactly.
Right.
It's more also, how do you deal with police? How do you deal with, most of us in our daily lives don't have to do this, but the moment you step out, all of this comes in, And so it's a very rich learning, but there is risk, wherever there is gain, there will be risk. So, yeah.
And the insects are very short-lived, isn't it? One particular insect life cycle may be completing in few weeks. So, and mostly these insects are very common in cold seasons, not in if the climate becomes dry, and if the scorching sun is there, then you don't see much of the insects. They will hibernate, or they will finish their life cycle. So how about for getting the information or data collection for one particular insect? What could be the average time we have to spend?
That's a very, very perceptive question. We have field seasons for every species. So some species only come up, like the Kudremukh species, come up only post-monsoon. So we can only work, sort of November, December up to April. Like you said, once it gets very hot, they have completed. So on average, for any given species, for the larger ones, only 3 to 4 months of the year. And for the smaller ones, there are 2 seasons. So you could get 6 months. But it's true. Our whole lives are driven by, and now with climate change, it's become so unpredictable. So it's quite difficult. You have to keep on going out and seeing whether populations are coming up or not. But again, it's part of the game, and you get used to living that way. The data. Yeah.
Thank you, Rohini, for introducing us to the music of the animals. It was a real
really
new topic for all of us. Yeah.
Thank you so much.
Yeah,
Very
Dr.
interesting.
Ahmed, before we leave, I would like to project today's evening talk that we have under Science and Our Life series. If you permit me, can I just show that? Because it's a very, very interesting topic, which I would like. I have posted the link also in the chat box. Can I just share my screen for a few minutes?
Yes, you can, ma'am. You can.
I'll just share the screen. Yeah. Are you able to see it? So today we are having a talk at 5 o'clock. by under the Science and Our Life lecture series by Dr. Abha Nagral. She is a very, very well-known, famous hepatologist, that is a doctor for liver. And she's going to speak to us on how do I protect my liver. And she has also been engaged in transplantation of liver at Jeslo Hospital and also Apollo Hospital at Mumbai. And if you, many of you are able to attend this talk, I think it will be really, really nice. So please, we all from IWSA invite all of you to attend this talk. So that's what I just wanted to tell you.
Okay, thank you, Lalita. Thank you, Khan. Shall we wind up?
Sure. Thank you.
Thank you very much.
Thank you, one and all. Rohini ma'am, Susan ma'am, Lalita ma'am, Aruna ma'am, Surekha ma'am. So all a big hippie hooray for all of us because we did a great job today. We have invited Professor Rohini ma'am. It was a bit, I'm going to say a good, eve today we have celebrated by listening to so many, sounds and though melody and noise is there, but a typical sound we can see that we could characterize today in the talk. So maybe it is a melodious in some way and maybe noise for some other people. But though these whatever insects are there, they live their happy life by having some communication signals. So thank you very much, Rohini ma'am, and all the Indian Women Scientists Association for supporting us for Department of Botany, Telangana University. So on behalf of the entire Telangana University and student participants who have attended this online virtual mode program, a big thank you for one and all. And with the permission of all, I would like to end the meeting now.
Thank you very much. Yeah.
Thank you, and see you again surely one more lecture.
Sure.
Thank you.
All right. Thank you.
Thank you.